So good morning to you all. As it was said, my name is Marisa Coelho, and the title of my PG project is Beta Blockers as New Therapeutic Options for Cancer. And just to tell you that this PhD research is under a co-supervision agreement between University of Porto and University of Insubria. And today I will present you some work that I've been doing during this research. And first of all, I would like to start by explaining you how the overall stress hormone response, how the sympathetic nervous system regulates the tumor microenvironment. And just to tell you that the overall stress response involves the activation of several body systems, including the hypothalamic pituitary adrenal axis and sympathoadrenomedullary system. And the effects of sympathetic nervous system on tumor biology are mediated by the release of catecholamines, which are released into tumor microenvironments to engage adrenergic receptors. So adrenaline is released from adrenal gland and circulates through vasculature to reach tumor microenvironment, and the noradrenaline is released from sympathetic nerve fibers into tumor microenvironment. And these hormones can have direct effects on tumor biology and tumor progression, enhancing the cellular proliferation, migration, and angiogenesis. And the adrenergic receptors, they belong to a superfamily of G-protein coupler receptors, and a total of 9 adrenergic subtypes have been identified and classified into 3 major subtypes: alpha-1, alpha-2, and beta. And today we will speak about these beta adrenergic receptors, mainly beta-1 and beta-2, which are present on all mast cell peripheral tissues where they activate intracellular signaling pathways in response to the catecholamines. But why are these receptors so important on tumor biology? So firstly, because they are highly expressed on cancer cell lines, and then because enhanced beta-adrenergic receptors expression and an overstimulation due to high levels of physiological agonists are involved with the hyperactivation of beta-adrenergic signaling pathway. So adrenaline and noradrenaline through beta-adrenergic receptors results in G-alpha-S activation of adrenal cycles and consequent synthesis of cyclic AMP. And the 2 main effectors of cyclic AMP in this context are the protein kinase A and the exchange protein activated by adrenal cycles. And these 2 pathways, they are able to induce transcriptional responses, leading for one side a upregulation of genes associated with the tumor progression and a downregulation of genes associated with the anti-tumor immune response. So this means that if we block this pathway using antagonists for these receptors, these, like beta blockers, these may play a beneficial role in cancer treatment. And actually, there are many epidemiological studies showing that cancer patients who are taking beta blockers for another reasons have higher survival and lower recurrence and mortality rates, which provide us, of course, a rationale for the use of beta blockers in this pathology. So the general aim of our project is exactly to study the influence of beta-adrenergic system on cancer progression, trying to understand which signaling pathways are involved and identify which beta blockers can be used to revert these effects or even have some protective effects per se. And in line with this, the first part of our project, in the first part of our project, we elaborate a narrative review in which we want to outline the influence of beta-adrenergic system on cancer cells proliferation, which is, as everyone knows, a crucial step in cancer progression. And we have the main aim to present and discuss the available preclinical and epidemiological evidences regarding the modulation of cancer cells proliferation by beta-adrenergic receptors, with a specific focus on the putative effects of beta blockers according to their pharmacology properties. And with this aim, we wanted firstly to clarify the presence of beta-adrenergic receptors, beta-1 and beta-2, on human cancer cell lines. And here I'm showing you the colon cancer example, but actually in our real table we have information about 10 different human tumors. But just to show you here that in colon cancer example, all of these cell lines express both adrenergic receptors. And then we want to describe the effects of adrenergic agonists and beta blockers on cancer cells proliferation. And here, once again on colon cancer example, we can see that all of these adrenergic agonists on the first column increase, from a general point of view, the proliferation of cells. And the beta blockers, when used alone, they don't change the proliferation or decrease, but they are able to completely revert the proliferation promoted by the adrenergic agonists. And the whole of this effect seems to be mediated by both receptors. So, from a general point of view, the main conclusions that we could get from the elaboration of this review were that both receptors are expressed in all cancer cell types, not only on colon cancer. Adrenergic agonists were able to increase the proliferation of several types of cancer, and this proliferative effect seems to be mediated by both receptors. However, in vitro studies exploring the underlying signaling mechanisms are lacking. And this is exactly one of our main aims, exactly to study the intercellular mechanisms underlying the effects of both adrenergic agonists. And with this proposal, we intend to evaluate the effects of these drugs, adrenergic drugs, agonists and beta blockers, upon different cellular dynamics parameters, namely angiogenesis, viability, invasion, migration, and proliferation, and try to understand in which way beta blockers can revert or have some protective effect. So, in line with this, our first, we start by studying the gastric cancer cellular models, and with this proposal, we evaluate the MKN45 from adenocarcinoma and K2-3 from carcinoma. And taking into account that we didn't have information from the literature about the behavior of these cells, we start by doing the growth cellular curves in order to understand when the cultures reach the lag phase, log phase, and the plateau phase. And then we wanted to evaluate the expression of beta-adrenergic receptors on K2-3 cell line, and as we can see here, this cell line expressed mRNA levels by which was evaluated by real time, and also protein levels, which were evaluated by flow cytometry. And regarding MKN45 cell line, we also proved that this cell line express both receptors at mRNA level and protein level. Then, in order to understand if the adrenergic drugs influence the cell cycle distribution in these cell lines, we evaluate by flow cytometry the cell cycle distribution. So these cells were treated with isoprenaline, which is a non-selective beta agonist, with propranolol, which is a non-selective beta blocker, and with adrenaline, which is the physiological agonist. And as we can see here, these drugs didn't influence the cell cycle distribution of these cells, which is consistent with the results that we got, that we obtained in our proliferation evaluation. Because as we can see, once again, these drugs didn't influence the proliferation of these cell lines. So the conclusions that we can get from these preliminary experiments is that these two cell lines, MKN45 and K2-3 cell lines, express both receptors at mRNA level and protein, and the cell cycle distribution and the proliferation of these cell lines was not influenced by the adrenergic drugs tested so far. So now we are trying to clarify if another cellular parameters and pathways are or not involved by beta-adrenergic signaling in these cell models. And for that, we are performing now angiogenic and migration assays. So finally, I would like to thank everyone from the Center for Research in Medical Pharmacology, in which I have been working over the last months, and also my colleagues from Porto, who also contribute to the publication of this narrative review. Thank you very much for your attention.